Om Amrteshwarya Namaha. A very pleasant morning to one and all. On behalf of Amrita Alumni Kochi campus, it is my immense pleasure and privilege to extend warm and heartfelt welcome to each one of you gathered here today and to those joining us online. Today's event is indeed a special occasion as it brings together the spirit of innovation, academic excellence, and the enduring bond of the Amrita family. We gather here for two reasons close to our heart: a scientific session by our own alumna, Dr. DHANYA NARAYANAN, and the release of the 11th edition of our alumni magazine, AMMAS ANUGRAHA. Before we proceed, I would like to convey the warm greetings and best wishes of our respected Group Medical Director and Provost, Dr. Prem Nair Sir, and Professor Shanti Kumar Nair Sir, Associate Provost of Academics and Dean of the School of Nanoscience and Molecular Medicine. Both are currently in the United States on official commitments and could not join us today. They have conveyed their heartfelt wishes for the grand success of this program. We are delighted to warmly welcome Dr. Girish Kumar KP, Principal of Amrita Institute of Medical Sciences. We also extend a heartfelt gratitude to Dr. PG Nair Ma'am, Emeritus Professor, Department of Pharmacology, Coordinator of the Nodal Office for Statutory Inspections, and Chief Coordinator for Campus Alumni, whose tireless efforts and vision have brought this wonderful event together. Dr. Beena KV Ma'am, Additional Medical Superintendent, Amrita Hospital, Kochi, whose gracious presence adds great value to today's program. A cordial welcome to Dr. Sabita Ma'am, Principal, School of Pharmacy, Amrita Vishwa Vidyapeetham, Kochi. A warm welcome to Dr. R. Venkatachalam Sir, Reader, Department of Public Health Dentistry, School of Dentistry, Kochi. We also extend a warm welcome to Mr. Kirtimon Sir from the School of Arts and Science, Amrita Vishwa Vidyapeetham, Kochi. To all our respected faculty members, alumni, students, distinguished guests, and participants joining us both in person and online, we extend a very warm welcome. Your gracious presence has made this gathering truly special. May I invite Sampujya Swamiji, Brahmachari Jayadamartha, who will now lead us in the invocation ceremony.
Guru Devo
Janaki Swamiji. In the absence of Dr. Prem Nair sir and Dr. Shanti Kumar sir, I now invite Dr. Beena KV ma'am, Additional Medical Superintendent, Amrita Institute, Kochi, to deliver the presidential address.
Namashivaya, and good morning to all of you. Just when I heard presidential address, I have decided for all the meetings, ma'am, I will prepare right from welcome address to vote of thanks. I don't know which role I love to play. So here from felicitation to promotion to presidential address, thank you for that opportunity. And at the outset, let me congratulate each person has got their own style of organizing thing. And I should call this event as PG ma'am style of organizing, because for the first time in the history of Amrita, I think we are having a Tuesday presentation of this kind with a prayer, seeking blessings of Amma, and then the other paraphernalias to follow, all these things. So hats off to you, madam, and congratulations for organizing in such a meticulous way. And see, usually for Tuesday presentation, they will just send a message and the topic that, and Diya and Dr. Pranav will sort it out and bring it as an all users bail. That's all. But here, I think almost since 1 month, Madam is after Diya for all these things, but Madam itself made all the arrangements, whatever is needed for this. And till this moment, Madam was arranging even for the prayer, lighting of the lamp. And so that means the commitment of Madam, and we should emulate from all these things that we have to be accountable for what role we have been assigned with. So first I'll also congratulate, see, alumni means weakness for ma'am, I think so. And alumni are, I should say that they are the ambassadors of that institution who carry all the values, the excellence and spirit of the institution to the world. Like that, here also the alumni, I think Dr. Dhanya is the alumni of our own institution, so we should be proud of that. In the master of ceremony, I was just heard saying that the bond of Amrita family will be strengthened by these kind of activities. So alumni means we feel that we are one among the members of the family. So here, Madam is under the leadership, I don't know who are all in the editorial board, Madam is bringing out the 11th edition of this alumni magazine named as AMMAS ANUGRAHA. Again, the magazine chronicles the achievements of the institution maybe, and also celebrates the milestones and inspires the future generation of our own family to get more involved in this kind of activity. And this particular topic, that is small, thinking small and treating smart, very catchy topic. And I wanted to listen to that, but because of my previous commitment, I may not be. Even then, I will get the recorded version and will definitely hear it. I was just asking Dhanya, what is this nano medicine? And I think nano, always small things and all breaks to me. We know that through the small, biggest innovations in medical field has come through small innovations. So that kind of nano medicine, I think it may be the precision medicine, the personalized treatment, and also better patient outcome and patient safety. That is all we are for. So let me wish this Tuesday presentation session on this particular, the next frontier in pharmacotherapy, very much needed for a clinical practice. And I wish the very best for this enlightening session. and also congratulate the entire team who behind this organizing this Tuesday presentation session, and also who has worked for the release of magazine, and hope that this session will be a very enlightening and informative session. Thank you, ma'am, for inviting me for this short session, and I hope that I have conveyed your vision of sharing this latest technology to the audience. Thank you.
Thank you, ma'am, for your kind words. I call upon Dr. P.G. Nair, ma'am, to present the magazine to Dr. Beena, ma'am. Thank you so much, ma'am. Nanomedicine represents one of the most exciting frontiers in modern healthcare. By harnessing science at the nanoscale, it offers the promise of delivering medicines with great precision, enhanced efficacy, and reduced adverse effects. We are honored to host our distinguished alumna, Dr. DHANYA NARAYANAN, Assistant Professor at the Amrita Center for Nanoscience and Molecular Medicine, with over 10 years of experience in translational pharmaceutical sciences and drug delivery. She holds a PhD in nano drug delivery and has led ICMR Department of Science and Technology and industry-funded projects in collaboration with IIT Madras and international partners. Her work spans microneedles, nano and micro drug delivery systems, implants, and RNA therapeutics. And she is an ISO 9001 lead auditor with great expertise in quality and regulatory practice. Ma'am will be delivering an insightful lecture titled Thinking Small, Treating Smart, Nanomedicine as the Next Frontier in Pharmacotherapy. Welcome, ma'am. Stage is yours.
Om Amriteshwarya Namaha. A very good morning to one and all present here. Thank you, Dr. Kirti, I guess the name is right, for the kind introduction. And I'm deeply honored to be here on this stage to give a scientific talk, because this hall holds a lot of memory to me as a student, as a researcher. And now when I stand here for a scientific talk, it's mixed emotions, okay. And I really thank PG ma'am for giving this opportunity to be here. And there are a lot of new faces, familiar faces. Welcome you all to this talk. And apparently, today morning when I walked into the hall, I saw the title, yes, it's here, of the Alumni magazine, AMMAS ANUGRAHA. Truly, that is very coinciding with what I have in my mind. It's truly AMMAS ANUGRAHA. I did not open the brochure PG ma'am had sent me last week, because I feel a bit shy to see my own picture printed on some brochures. Okay, so I didn't open it. I did not even open the email also, which actually had the, Tuesday presentation email. I did not even open that. But when I walked in today to the hall, seeing my picture everywhere was slightly embarrassing. But then my eyes was on AMMAS ANUGRAHA. So I'm deeply, grateful to Amma for giving this opportunity to be here. And whatever I have today is given to me by my gurus. So I'm so happy to impart whatever I know to the future generation. May it help you. And PG ma'am was, I have to mention, like Beena ma'am told her in her presidential address, she was very specific about the talk, that it should be useful for the upcoming generation. And I have not seen any organizer taking so much of effort in conducting this. You'll not believe, she in fact checked the font sizes. She was so particular about that even the last bencher should be able to see that. That actually shows her commitment. And a lot to learn from you, ma'am. Thank you. So without wasting too much of time with the introduction, let's start with a scientific session. The title is Think Small, Treat Smart. and nanomedicine as a smart frontier. Okay, so I'll just give a clinical scenario. Okay, let us start with the world's most deadliest disease, which every researcher is trying to find a solution for. Can you guess what that is? Cancer. Yeah, yes, cancer. So before starting, let this be an interactive session, okay? Otherwise, it would be boring. Early in the morning, sitting, just a monologue will not work. Okay, so cancer, let us start with cancer. Imagine a clinical scenario wherein a person is diagnosed with cancer today. What are the three things that you expect with the current clinical background that we have? What are the three things that a patient would face? Any guesses? Starting with diagnosis. Either it would be a late diagnosis, right? Why? Because the diagnostics tools that we have right now in clinics, they are not sensitive enough to capture the biomarkers, which are very less in quantity at the early stages. True? Yeah. And the second stage, when the patient starts taking medicine, what is happening? Is it reaching the target? The entire, suppose 50 mg of the drug molecule is given. Will that 50 mg reach the target? Suppose that patient is having breast cancer. Will that 50 mg reach the breast tissue? No. So what is the result of that? You have to frequently administer more and more amount of drug, right? And what is the result again? Drug-induced toxicity. Toxicity to the unwanted organs. Okay? So these three things. So do you really think now the challenges that we have in clinics, is it because we don't have a diagnostic tool or we don't have drug molecules in practice for clinics? Is discovering new drugs more important right now, or giving the existing drug molecules in a right way that is more important? Situation 1 or situation 2? Respond here. 2. Because discovering a new molecule will take off easily 10 to 15 years from your life. So what about the patients in that lifespan? They have to suffer. So each time, discovering a new molecule is not the solution. It is giving the right molecule at the right time, at the right concentration is the solution. So do we really have a solution to do so? Yes, we do have. So this is Richard Feynman, father of nanotechnology, whose vision was ahead of his time. And he said, there is plenty of space at bottom. Yes, think small. Go, think of each atom, each molecule that can be of help. There is plenty of space at bottom, and yes, you can do wonders with the existing therapeutics. And that's where nanomedicine has a critical role to play. So why do we need nanomedicine? So what are the current challenges that we have with the therapeutics right now? Either poor solubility. So what happens if the solubility is very poor? Rapid clearance or accumulation into the organs. So before getting into what exactly or how exactly nano can be engineered, going into the nanomedicine, let me ask you a question. Was this a human invention? Nano? Any guess? You can even guess. I don't expect a correct answer from you. I know most of you are new to the subject. So was nano a human invention? No. Let us look into our body. This is the scale of life. Many molecules work at nanoscale. The genetic information, DNA, stored at nanoscale. Many proteins, antibodies, molecules, everything is at nanoscale. Now, when you go look at the nature, have you heard about the lotus effect? What is lotus effect? What happens if you drop water molecule, water onto the lotus leaf? Will it get spread? No. It would roll on the surface. Have you ever wondered what the reason behind that is? So the scientists went into the deep search of what this lotus effect is. Why the water molecule is not spreading out on a lotus leaf. Why it is rolling around. That's due, you see that bumps. This is the electron micrograph image of the lotus leaf. These bumps have waxy protrusions. So these bumps are around 10 micrometers, and this is around 100 nanometer protrusions, waxy bumps. This make the lotus leaf completely hydrophobic. What is hydrophobic? Water repellent. Okay? It cannot hold water. It cannot absorb water. So as a result, what happens? The water rolls on the surface. And scientists were inspired by this nanotechnology wonder from the nature. And they designed these medical gauzes which are antibacterial and water repellent. Now we have this in clinics. The next is the butterfly wing. So you have seen many butterflies, right? This is a morpho butterfly. Why does it have a blue colored wings? Is it due to any pigments? The blue color is that due to pigments. So whatever colors you see in nature, some or the other pigment is behind it, giving the colors. So there is a change in this morpho butterfly. It's not any pigment that is imparting the blue color. You see that electron micrograph image wherein there is 200 nanometer chitin arrangement. So when sunlight falls on it, it reflects a particular wavelength of light which is blue in color. That is why morpho butterflies' wings are blue colored. Again, the next thought of scientists, they wanted to take this technology, get this into some of the other useful things. Okay, now many optical biosensors and ophthalmological implants were made depending or taking up this technology. So what is the lesson from this? That nature has nanotechnology. Nanotechnology is not a new thing but existing in nature. We are only going deep into the technology and understanding if we can take that technology up, understand more and more, and use it for the improvisation of patient life. Okay? And to use it for the patient life in clinics. So I am again remembered by a quote by Richard Feynman, father of nanotechnology. The more and more you know about a subject, the more and more ignorant you become. Have you ever felt so? I have felt. I was asked about a question. I was asked this question after my PhD defense by the committee. What do you feel now? You have completed your PhD. What do you feel now? The answer that came from me was, I feel I don't know anything. I'm only starting. That was my feel, and that's my feel right now also. It's a very familiar subject to me, and I wanted to, give 100% justice on today's topic. I was reading lot of confusions came up. Okay, so even if you get confusions, it's quite natural. Don't worry. So we can clear it up during the session or after the sessions. So if you have to use nanotechnology, you have to know what nanomaterials is. A little bit of fundamentals. What is nanomaterial? So by now you know what nanomaterial is. We know the scale. I showed you a scale. Here also you have a scale. So here you see a scale. Why do you need a scale always? There was a puzzle in our school days, right? A question which often all of you might have face that question. Your friend might draw a line, okay, and ask you to make that line smaller without rubbing off. How do you make that line smaller? Draw a bigger line on its side. Then naturally this will become smaller. So you always need a comparison to understand how small or how big your material is. So when you look at this, it starts from 1 meter to 1 arm string. Okay, so our interest is on this blue area, 10 power minus 10 to 10 power minus 9. That is 1 billionth of a meter. Don't get confused about numbers. For ease of visualization, let me tell you, consider Earth. We are all familiar about Earth, our planets, right? 1 meter is Earth. You place a marble next to Earth. That marble is 1 nanometer. It's that a difference between 1 meter and 1 nanometer. Okay. So, by definition, it says we have to have the nanomaterials within 1 to 100 nanometer. But in practical applications in biology, we don't restrict it to 1 to 100 nanometer. We use up to 400 nanometers. And why nanomaterials are different? Why it behave differently? So the properties, the entire properties are different when the scale is brought down. Okay. The 3 major contributors on why the nanomaterials behave differently: the exponential surface to volume ratio. I'm not taking you through physics. I know all of you are biology students. Okay? Just imagine you have a solid sugar crystal and you have powdered sugar. Which one will dissolve fast? Powdered sugar. The same molecule, the same content, the same chemical, everything is the same. But what happened when it was powdered? The surface area got increased, the surface atoms got exposed, the reactivity increased simply. So it was rapidly soluble. That is what is known as surface to volume ratio. Quantum effects. Interested in going to quantum mechanics? Even if you are interested, I am not. Okay? So quantum effects is nothing but the changes in optical, magnetic, and electrical properties. Okay? When the size is reduced, there will be changes in electrical properties, magnetic, and optical properties. So, what is the color of gold? The gold that you wear? Golden. But what color? Metallic yellow. And what would be the color of gold nanoparticle? Yes? Yellow? No. The gold nanoparticle solution looks red, purple, or blue according to the nanoparticle size. That's because of a phenomenon known as surface Plassmann resonance. Let's not go deep into it. Again, it's the atoms that is getting exposed and the oscillations when light falls on it. Okay. So the property, you just understand the property changes. And this color change is being used in diagnosis field. Okay, you use gold nanoparticle of different sizes. When it is 20 nanometer, it is red. When it is 40, it is purple. When it is 80, it is blue. When it aggregates, altogether it changes. So this property is used in diagnosis. Okay? And the third one, biological interaction. The most powerful thing that a nano can do. Just because it is tiny. No hefty molecule can do this job. Just because the nano is too small, it can interact with our biological system just like any other biological molecule in our body. No interference with any other, components in the body. There are some things that you have to take care when you design nanomaterials, which I'll be discussing in the further discussions. Okay. So the 3 critical things that you have to keep in mind on what happens when a material is changed to its nano form from a bulk form. Okay, and these 3 things are majorly explored for any therapeutic or diagnosis purpose. Those are the 2 major arms in clinics. So today's topic, we'll be concentrating more on healthcare. Nanotechnology is used in many other fields, But since we are a healthcare campus, let us concentrate majorly on diagnosis and therapeutics. So just for your information, other than this, the nanomaterials are classified based on its shapes and sizes as 0-dimensional, 1-dimensional, 2-dimensional, and 3-dimensional, just on the basis of how many dimensions falls to the nanoscale. If all the 3 dimensions fall to the nanoscale, it's 0. And if 1 dimension is extended apart from the nano to a micro, like the tubes you see here, those might be slightly micrometers or sometimes millimeters, but the other 2 dimensions would be in the nano range. That is 1. Similarly, 2 and 3. So when you see this 3D structure, the scaffolds or the textile, nano-textile sheets that you make, you might get confused where nano is coming up. So here, the features are at nanoscale. The pores or the materials that you use here are at nanoscale. You use the nanocomposites here to impart the better features or enhanced interaction with a biological scenario. So this is all about the fundamentals of nano. Now we have to apply this to the healthcare. Where can we apply this? So on the whole array of current medicine, you can apply this nano. Starting with diagnosis, imaging, therapy, recently vaccines, not recently, from last few years, the studies on vaccines are going on, and regenerative medicine. So, as I told earlier, let us concentrate more on the diagnosis and therapy side. So, nanotechnology in diagnosis. So, we had a prelude wherein we talked about a clinical scenario, right? What were the challenges we had in diagnosis? Low sensitivity, low specificity, low image resolution. Okay, so the researchers have solutions for all those. So here I have listed down the challenges, and these are just 3 works that I have taken from literature. There are many research works currently in the literature, but 3 that I found which was very interesting is being taken. So this is a zinc oxide nanowire matrix. There are millions of nanowires here. So what happens when even at the early stages where you have very little biomarker quantity in your sample, biological sample, 1 or 2 biomarker interaction would change the electrical property. This electrical property will be captured by the sensors. So it is that sensitive. This is a nano chip wherein you have a lot of antibodies embedded on it. And each antibody has been tagged with a barcode. Are you familiar with barcode? When you go to supermarkets, whatever products you take, at the billing, they'll scan something, right? A barcode. Similarly, there is a DNA sequence. Let us call it as a barcode. And this would help the biomarker to specifically bind to the specific antibody. Specific. So that increases the specificity. And the third one, I'm not sure if you can see the image clearly. This is to again improve the specificity and the image resolution, wherein you have the nanoparticle loaded with contrast agents, for example, gold nanoparticle. Okay? And these nanoparticles are decorated with specific proteins, such that the cells that are overexpressed, I don't know if you're familiar with the term overexpressed, you check for certain biomarkers. For example, in breast cancer, HER2 is a biomarker. So that is overexpressed. You check if that is highly expressed. Okay, so you tag this and send it to the cells such that it goes specifically to that particular cell and get internalized. Okay, it will not go to any other cell. It is specific to that cell. Okay, it's a ligand-receptor interaction. So you have the ligand on the nanoparticle and it specifically go and bind to the receptor and the receptor takes in the particle. So have you learnt about the endocytosis, wherein some molecule go and attaches to the cell that invaginates and then takes in as a droplet? Similarly, whatever receptor, whatever molecule go and bind to the receptor would internalize that into the cell. Okay, and that green dots you see here are the nanoparticles that have been internalized. Why are we able to see this? Because the image resolution is very high. They are specific to the cell type. So there is no matrix interference playing a role here. Okay, so this is another moiety or modality that I found very interesting during the literature search. It's a triple modality nanoparticle. Okay, now during diagnosis, we have to have many contrast agents for many imaging techniques. One for MRI, one for PET-CT. You're all aware about that. But in this nanoparticle, you have different layers. Okay? Just by injecting this nanoparticle in the body, any imaging starting from the pre-surgery, that is planning of the surgery, to the post-surgery confirmation, if the entire tumor has been taken off, can be done. Like, the MRI, photoacoustic, Raman imaging, okay? Because of the different components in the single nanoparticle, that is triple modality. So here we have the gold core that would give you a clear image of how deep-seated your tumor is. And this Raman imaging, this Raman active layer, will give you an idea of the tumor margins. Identifying tumor margins is very critical. Do you know why? If some cells are left out on the margins, there are chances, high chances for, yes, the tumor to come back. Okay, so, sorry. So this gives you the single nanoparticle will walk you through the entire procedure, starting from pre-surgery to the post-surgical confirmation. That's about diagnosis. Let's move into the therapy. Therapy is equally or more important. Just by diagnosing, you cannot do anything, right? You know that, yes, I have this. But how do you treat it? That is also very important. So, some points were discussed in the beginning. There are many things nanoparticle can do in therapy. Okay? So, the first thing, nano carriers enhance drug solubility and prolong systemic circulation. Why do you actually need to enhance solubility? Because now, in clinics, solubility of the drug is a critical challenge. Okay? Just because the drugs are poorly soluble, it's just a cartoon image that is shown for ease of understanding. What happens? A rapid clearance, and the result is low bioavailability. So again, when it is poorly bioavailable, what is the output? Frequent administration. Okay, it's not bioavailable, the plasma concentration has come down, the next injection or the next tablets. Again, the same thing would happen. You're not working on the molecule, you're just working on the dosages right now. So is there anything that we can do about this molecule without changing the chemical property? Because if we change the chemical property, then the efficacy or the outcome would change. So we don't want to touch on the chemistry part of the molecule. So without doing anything, can we enhance the property of a poorly soluble drug? Yes. We can encapsulate this in nanocarriers such that it becomes dispersed in blood. Okay. This blood, the major component is water. So our idea is to disperse this in blood. Do I need to go fast? We have time, yeah. Okay. And these protrusions that we see here, you're very familiar about PEG, right? PEGylation. So this is a hydrophilic polymer. So what does it do? It makes a particle stealth. What do you mean by stealth? So immediately when you inject any particle system into your blood, systemic circulation, what is the first thing that happened? It is identified as a foreign body. So what happens? Our immune system would trace it. Okay, that is the action, that's the immune system, border security force that we have. If it is not working that way, there is some serious issue with your body. Okay, so a lot of proteins come and bind. And the proteins named as opsonins, opsonization is the term for that, okay. If your particle is getting attached by opsonins, that gives eat me signal. So immediately, the macrophages from liver and spleen come, eat up the particle, and is cleared off from the system. But by PEGylating this nanoparticle, the proteins are avoided by the steric hindrance property. The proteins will not come. So this is a cartoon showing that. So if there is no PEG, complete protein, adherence, okay, it adsorbs. And with PEG, there is no protein, and it prolongs the circulation. So since it's an alumni talk, I thought I'd show you this with a real result from my own PhD work. So here, I worked with a non-steroidal anti-inflammatory drug, okay? So that has poor bioavailability, a short half-life, many issues, but that's routinely prescribed in clinics. How to improve this medication? That was the goal, entire goal. So I had to nano-encapsulate. So when you see here, sorry. What happened? So by the time that is rectified, so when you see here, you see 3 lines. The green one is a bare drug without any modification. So you can clearly see the timeline, it clears off by 5th hour. And this blue line is a clinically administered formulation, that is Vovran, which gets cleared off by 48 hours. And the red line is the nano-encapsulated formulation that we developed at our center, which clearly goes up to 6 days. It clears up by 6th day. So it is a PEGylated nano formulation. So clearly, by seeing the area under the curve, you know that the bioavailability is enhanced by nano-encapsulation and PEGylation. This was further validated by the pharmacodynamic, the animal model, okay? The efficacy. A paw edema model was developed, and the formulations were tested. So you can see 2 sections, up to 6 hours and 24 to 96. So up to 6 hours, you see Vovran or, the diclofenac sodium or the nano formulation, everything has similar activity. Up to 6 hours, the edema was under control. But after that, the edema was, higher, and it came back to its treated stage, wherein edema was induced for the other formulations except for the nano, wherein edema was well under control, which means the doses that it was releasing was imparting therapeutic efficacy. So with very little dose, with a single injection, you could control inflammation for 6 days. This is our own study, okay? And the next thing, so the previous slide talked about improving the solubility and enhancing the circulation time. Now the second thing the nano carrier can do is maintain therapeutic levels for longer duration. Though we saw it in the earlier graph, I have to mention this because it is important. All the toxicity issues or the administration issues, patient non-compliance comes due to the rapid clearance. So here you can see this graph has a peak and then a valley. Okay, this peak and valley is a major issue when we go with the hormone treatments. The patients cannot suffer the peak and valleys. Any other treatment also. So when there is a peak, it might go up to the toxic level also, which would be highly, toxic to the organs for a patient. So what can we do? Again, when you encapsulate due to the matrix properties. Okay, here the matrix properties become important. So when I say matrix, it is the carrier that you use to encapsulate the nano drug. It can be a polymer, it can be a lipid, inorganic, organic, okay. So whatever it is, it majorly depends on the matrix characteristic that it can either diffuse, degrade, swell, or based upon any stimuli, it can release the drug molecule slowly from the carrier system. So what is the result? You get a sustained release. Okay, so I'll show you a graph from the literature. So a nanoparticle was given. So here, the blue peaks and valleys you see here are individual administration, conventional release, wherein you need 4 administrations to keep the therapeutic levels. But for the nanoparticle with a single injection, the therapeutic level was maintained, and this 4 administration could be replaced with just 1 administration. The frequency of administration can be reduced. In our study, during my PhD, I observed that the bioavailability was enhanced by 56 times, okay, when you statistically calculate it. So it is that powerful nanoverse. The next point is nanocarriers minimize toxicity. We covered this topic. Why it minimizes toxicity? Now you should be able to answer. There is no burst release, okay, because it is protected in a shell. It is not immediately available to the body, such that it is rapidly distributed to all the organs, okay. It is confined in a nanocarrier system. So only the wanted amount that we have to design according to this release kinetics that we need, the release is controlled and the toxicity is controlled. So there is another thing which I'll be talking about, the passive and active targeting, which can also control the toxicity. And this is widely used for cancer therapy nowadays. Okay. Before that, it's again from my PhD thesis, how the toxicity was reduced. So hypersensitivity and thrombophlebitis for diclofenac sodium is widely reported. So here, I don't know if it is very clear to you. Do you see piloerection? The hairs standing up. It's a hypersensitivity reaction immediately when diclofenac sodium was injected to the rat. And here, the rat is normal, wherein a nano formulation was injected. And this was intravenously injected through the tail vein. Okay, these 3 tails, the first one is the control saline solution, the second one is just the diclofenac, and third one is a nano formulation. So you see a thrombophlebitis. Tissue damage is very extensive for diclofenac sodium, which was well controlled when we nano encapsulated it. It was safe, it was not toxic. So this is about the mechanism to improve the drug accumulation into the tumor tissue specifically, but it can be used in many other diseases also. So there are 2 mechanisms. One is passive and one is active. So by the word passive, what does it mean? Something in your body. You're not doing anything extra to the carrier system. Something in your body is helping you to drive the particles to that particular area, the target of interest. So what is happening in the cancer tissue? So the cancer, it's a characteristic feature of cancer. So we have to keep this in mind whenever we design the carrier systems. There is a leaky endothelial system. You have gaps between the endothelial cell lining and poor lymphatic drainage, due to which there is a property, I don't know, I have mentioned it, EPR, enhanced permeation and retention. Enhanced permeation is due to the leaky vasculature and retention is due to the lymphatic drainage problem. Okay, poor lymphatic drainage. And in the case of active targeting, again it's a ligand receptor interaction. You modify your carrier system with specific ligands, you send it to the body, and that ligand specifically identify the receptor and your molecule is taken into the target cell. Okay, that is about active and passive targeting, wherein you can increase the drug accumulation. Now, is it just a fiction? Is it all possible? would be your question. Yes. And we have drugs in markets. I'm not showing the entire list of drug molecules. Up to date, I think there are around 90 nanomedicines in market right now. But let us take, the unforgettable nanomedicines. The first nanomedicine that was approved, doxorubicin. It's a chemotherapeutic drug. It had high cardiotoxicity issue. And the cardiotoxicity issue was due to the rapid distribution of the drug molecule to the cardiac tissue. Okay. So this rapid distribution was controlled by encapsulating this drug molecule in a liposome. And on top of liposomes, there was a PEG corona that was given, which increased the circulation time. And thus, the heart was saved. This is widely used in clinics. And the first protein nanomedicine, protein-based nanomedicine, Abraxane, paclitaxel. The problem that the conventional paclitaxel had was the solvent, a toxic solvent, because the drug is poorly soluble. So they had to use a toxic solvent, chromophore, which led to hypersensitivity reactions and many nephropathic problems. And the solution the scientists found was encapsulating that in a native protein, albumin. So there is no issue of clearance. Albumin is found abundantly in our body. Okay, so encapsulating paclitaxel with an albumin. Again, in the earlier case, doxil, it was enhanced permeation and retention, EPR effect that was exploited. Why? The size was critically designed to 80 to 100 nanometers such that it can pass through the leaky vasculature. Here, since it is albumin, it has albumin-specific receptors on our cells. Okay? So it is a combined technique of both active and passive targeting. The revolution. What happened during COVID, SARS-CoV-2 outbreak? For scientists who worked in Nano, this was a global validation. Yes, Nano works. And Nano saved millions of lives. And there is nothing more for me to talk about the COVID vaccine. Anyone of you who has not taken a shot for COVID? All of us. And all of us read a lot about the vaccines, nano vaccines, out of fear. Not to know more. Yes, out of fear, we all read a lot and we gained confidence because it saved our lives. There was a diagnostic tool also that was developed that used the gold nanoparticle. You remember the optical property that I talked about, the gold nanoparticle? It changes, the optical property changes with a change in the size. So when the biomarker was introduced to the gold nanoparticles aggregated, and there was a color change within 10 minutes to blue color. If it is positive, it would change to blue color. That was a diagnostic tool. And here, the vaccine, the mRNA was safely secured into the LNP systems. The Pfizer and Moderna, the two companies, used the LNP system, whereas the Covishield used a viral vector system. But the reports suggest that 95% efficacy was imparted by the LNP systems, liposomes. The reason is our body has liposome. It can well integrate into the cells. And it is safe, scalable, and there were many variants. You remember variant 2, variant 3, there were many variants for COVID at that time. So it was easy to manipulate the system for LNPs, where viral vector system had many manufacturing difficulties. Okay, so LNP systems was, a global validation was done for the nanoparticle systems during COVID. Now, where are we heading to? The smart nanomedicines. This is a cartoon depicting how a nanoparticle can be designed to release in response to a stimulus. Okay? So here in the bloodstream, you see a nanoparticle which is in the locked state. When it reaches inside a cancer cell, it opens and releases the drug molecules. So this is a particular stimuli responsive system specific to the pH. You have acidic environment inside the tumor microenvironment. So you use the polymer, the carrier system that is specific to get released into the acidic environment. So it opens up only when it reaches that acidic environment. So the scientists are using many other responses like temperature, glutathione concentrations, hypoxic conditions wherein you have a low oxygen content. So many stimuli responsive nanomedicines are under development. The next is theranostics. Theranostics. What is theranostics? We talked about diagnosis, we talked about therapy. What is theranostics? Therapy combined with diagnosis. So you can use a single system, a multifunctional nanoparticle, both for diagnosis and for therapy. So you'll have a contrast agent in the nanoparticle, you'll have a therapeutic moiety in the nanoparticle, which is targeted, and you can easily identify where it is getting localized, and when it is localized, if it is a stimuli-responsive molecule, polymer, it releases upon the stimulus, or releases as per the matrix's nature, as we discussed, either due to diffusion or erosion. Okay. This was interesting when I read. It's about nano-informatics and nanobots. Okay. AI. This is AI era. Nothing works without AI, right? So there are many advancements that can be done in nanosystems along with AI. You see, you can predict the systems. You can predict how your systems work. I really wish if it was there 12 years back, because it took 3 years for me to optimize the formulation. I had saved probably 2.5 years if AI was there. Okay, so this is the system which can predict your formulation. It will save your time. It will save patients' time too. The faster it comes to the market, the more beneficial it is. And this is about nanobots, okay? You have heard about origami, right? What is it? Craft. How many of you have that origami hobby, wherein you fold the papers into many beautiful things? So this is DNA origami, okay? Wherein you fold DNA itself, okay? And you send it with many commands and or logic commands. So how does it work? It will release the therapeutic moiety only if there are 2 molecules, like biomarker A and biomarker B. Or it can release if the command is like biomarker A or biomarker B. Why this is important is because not all the cancer types are same. Okay, for the same cancer, for the same patient, 2 patients who suffer from breast cancer, for one, the medicine might work. For the other one, the medicine might not work. Why? Because the reason why one got cancer is different from the other. Okay, the marker that would be expressed would be different. The underlying reason would be completely different. So one short medication will not help. That is where personalized medicine is very important nowadays. You identify what exactly the problem is and you tailor-make the medicines. Okay. Now, I've not included personalized medicine here because that has to come along with molecular medicine also. Probably in some other sessions, if interested, we can cover up. Because here it was only nanotechnology that was covered. Okay. That is why personalized medicine is very important. And this nanobot is a future promise. So before I stop, let me show you the products that were developed. I mean, few of the products. There are many, many products that are developed in the last 20 years. These products, the stents, the dental implants, the bone grafts, okay, all this has completed the phase 1 clinical trial. This is all for regenerative medicine. And in the therapeutic part, for glioma, we have an implant system loaded with drug molecule. We have an injectable gel system. This is very interesting because the reason why a patient, the survival period is 6 months for glioma, is because there are deep-seated tumor tissues, I mean cells, cancerous cells in the brain. So whatever the surgery or the implant system will not be able to reach the deep-seated cells, cancer cells. Okay, this injectable gel can travel up to 3 to 4 centimeters inside the tumor tissue, and that can kill the cancer cells. So that is much more, and again, non-toxic, releases the drug molecules very slowly. Okay, so it can control. Only the preclinical study has been completed here. And to summarize, the human future we can do more with less. So the next time, so what are the things to make it into a nutshell? We can reduce the cost, reduce the waste, and it's equally accessible to everyone. We can make it equally accessible. So next time when you hear about Nano, it is not just the small picture that should come into you, it should be the power that Nano has. Think small, treat smart. Thank you. I would be happy to take up questions, if any. I know all of you might be hungry.
When you do regulation, the drug effect will not come down?
No, ma'am. That has property only outside the carrier system. Your drug molecule is inside the carrier system. But I skipped one point. PEG is not the wonder molecule anymore. There are many toxicities reported with PEG. The patients develop antibodies against PEG. So there are many other hydrophilic molecules that are into research, which can be used instead of PEG in a much safer way. Why the COVID vaccine induced hypersensitivity reactions in few patients? That is because the antibodies that were previously generated antibodies against PEG. Okay?
Yeah, thank you, ma'am, for the insightful. Are there any toxicity associated with the nanoparticles first?
Yes, we have to carefully, that is why selection of material, the selection of the synthesis route, everything should be carefully curated. The designing of the nanoparticle itself is a different topic. So you have to carefully select the approved nanomaterials at the approved concentrations with, reduced chemical solvents in it. And then remove the solvents, test the residual solvents, if any, and check the safety. You have the entire safety toxicity evaluation guidelines available. So your system has to pass that safety and toxicity studies. Only if it passes that, you can give it to. So because this is given mostly into the systemic circulation or by oral, the chances for the particles to, even if you have the hydrophilic corona on top of it, there are chances that particles can accumulate. So what happens when agglomerate? What happens? The size increases. When size increases, it can accumulate into organs like liver or spleen. So if the accumulation is more, it can be toxic to the tissues. So you have to be very careful about the design.
And will there be any immune response against it because it is also a foreign particle?
That is what the selection is critical there. That's why the FDA or GRAS approved materials play a major role there. Okay. In immunogenicity, there is a panel of tests to be conducted. So immunogenicity is one test that has to be conducted. Any foreign molecule, when it gets into the body, you have to expect some kind of immunogenic reaction. But at nanoscale, the particles or the systems that you design for therapy should not induce any immunogenicity. But there is another section now, immunotherapy, wherein you modulate your immune system to act against the disease. There you have to induce immunogenicity. So it is according to the requirement that we need.
Thank you, ma'am.
Yeah, I would be happy to take your doubts even at my office or outside during coffee break. I hope I did justice to my job. Thank you for the opportunity, and thank you all for your patient listening. Thank you.
Thank you, ma'am, for that insightful and enlightening session on nanoscience in pharmacotherapy. Your clear explanations and thought-provoking perspectives have deepened our understanding on nanoscience. May I call upon Dr. P.G. Nair, ma'am, to hand over the memento to Dr. Dhanya, ma'am. May I invite Dr. Divya ma'am to present the certificate to Dr. Dhanya. Today's program is made even more memorable with the release of the 11th edition of the Amrita Alumni magazine, AMMAS ANUGRAHA. More than just a publication, this magazine is a reflection of achievements, memories, and continuing journey of our alumni community. It beautifully captures the spirit of excellence, service, and lifelong learning that defines Amrita. Dr. Giri sir is unable to join us today as he has been unexpectedly held up with an important commitment. May I now invite the esteemed dignitaries on the dais to release the magazine and to receive the first copies. I invite Dr. Sabita ma'am, Principal of Pharmacy College, to release the magazine. I also invite Dr. Ramakrishnan sir from Amrita School of Medicine, Dr. P.G. Nair ma'am representing our campus, Dr. Venkatachalam sir from Dental College, Mr. Kirti Mon sir from Arts and Science College, Dr. Dhanya ma'am from Nano Science Department, Dr. Prini ma'am from Nursing College. I also invite Dr. Prasad sir from Allied Health Sciences. From Arts and Science College. Congratulations to the entire editorial team on this milestone. Thank you all. I now invite Dr. Adra from Department of Pharmacology to propose the vote of thanks.
Good morning, everyone. On behalf of Amrita Alumni Kochi campus, I extend our heartfelt thanks to everyone who made this morning special. Our sincere thanks to Dr. Prem Nair and Professor Shantikumar Nair, both away in the United States and unable to join us, yet warm in their wishes for this event. We thank Dr. Beena ma'am for presiding over this function. I extend my sincere gratitude to Dr. P.G. Nair ma'am, the organizing head of today's session, for her guidance, vision, and efforts in making this program possible. Your dedication to fostering academic discussions and learning opportunities are truly appreciated, ma'am. Our deepest gratitude to Dr. DHANYA NARAYANAN ma'am for an enlightening session generously shared with us. We also thank Dr. Savita ma'am, Mr. Kirtimon, Dr. Ramakrishnan, Dr. Dhanya ma'am, Dr. Prini ma'am, Dr. Prasad sir, and Dr. Venkatachalam sir for gracing us with the magazine release. We are grateful to our alumni, faculty, and friends here in the hall and joining online for your presence and warmth, and to everyone working quietly behind the scenes, including the audiovisual team. Thank you so much for making this morning possible. Once again, thank you, ladies and gentlemen, for being with us this morning. Thank you so much.
Thank you, Dr. Adra. This brings us to the end of this memorable morning. We warmly invite you all to join us for the breakfast served just outside the hall. Thank you for being with us today. All the very best.